I'd like to introduce Rob Cannings. Just going to give you a bit of a bio for Rob. Rob joined the RBCM in 1980 as Curator of Entomology and retired 33 years later in 2013. From 1987 to 1996, he managed the Natural History Department in the museum. He is now Curator Emeritus and continues to work on research projects and play in the insect collection. He has spent a lot of time studying dragonflies and robber flies. He's published widely on many insect groups, from mantid flies and moths to lamprey beetles and bumblebees. But in retirement, he's concentrating on robber flies. He served on the scientific committee of the Biological Survey of Canada, the Arthropod Subcommittee of COSIWIC, which is the Committee on the Status of Endangered Wildlife in Canada, and the British Columbia Invertebrate Recovery Team. He is also a past president of the Entomological Society of BC. Rob got his Bachelor's of Science from UBC in zoology, his Master's at UBC in zoology, and his PhD at the University of Guelph in entomology. Over to you, Rob. You've got a solid 35 minutes, and take it away.
Thanks, Leah. I don't think it was worth the amount of time that we took to introduce me, since, as my wife said this morning, the less you talk, the more successful this will be. She also said my main purpose this morning was to keep Gavin under control. So, yes, we are dependent entirely in this experiment on questions from the audience. This is the purpose of this. That's why we forbade you to ask any questions after each of the little talks. So please ask away, and I will be quiet.
I'd like to know when and where your next BioBlitz will be.
Are you, when and where the next BioBlitz will be? The insect spider BioBlitz, or BioBlitzes in general?
In general,
Yeah.
Yeah.
Well, of course, I'm not sure, so I'm gonna ask Claudia or anybody else to.
I suspect the next BioBlitz might be in Machosen, because they've been doing an annual BioBlitz, and so that's nice and close. I haven't heard of another specifically, but I know I have the Whistler BioBlitz, which is really fun.
Yeah, and there's also
Hackeye, yeah.
There's also beginning 2 or 3 years ago, an annual BioBlitz somewhere in the Yukon. And I have to say that the Yukon has sort of partnered with us in many disciplines because we consider them a colony of BC. And so there is a BioBlitz up on the Dempster Highway this summer.
And just to underline the value of BioBlitzes in general, I mean, it's something that Claudia and I both referred to that are really integral to what we do. I mean, it's a chance to work with the public. And it is, I mean, to some degree, it's an outreach activity. It's our chance to go out and say, oh, look, these are great plants or great insects that we see. But it's a chance for us to get specimens from unusual places. And more than that, it's a chance to really work with communities, because a lot of them are driven. You know, Machosen is driven by community efforts. I worked on Valdez Island, which was by community efforts working with the Liaxin First Nation. And working on Galiano, a lot of these are local people that want to know what's there. They bring us in, they give us lunch and say, help us to know what insects and plants are there. So the BioBlitz idea, I think, has really revolutionized how collections are built in a modern way, because it's a better chance for us to get there, and plus it has more meaning to the people that live there.
Ange.
First of all, that's really loud. Sorry about that. Everybody awake? First of all, I want to thank you all for your presentations today. I learned something new every day. I've been here 15 years, and every day it's like marvelous. I am really curious about partnerships. You all spoke about partnerships and the value of partnerships in completing our work, completing your research. And I'm just curious how partnerships come to us. Do you guys go out and find them? Do people come to you? I'm sure all of you can talk a little bit about that. I'm just really interested. Go ahead, Henry.
Is this one? Yeah, it's working. It's a bit of both for me. Fisheries and Oceans have had a long-standing interaction with the museum, and so I'm just joined in on that. So yeah, I've helped culture that. But the connections down to in the United States, Moss Landing Marine Laboratories, that's a new one, and that came out based on collaborative work with the DNA barcoding project, because the Moss Landing Laboratories realized that the barcoding initiative had made mistakes, and so they asked us to verify what we had, and so UVic also is a long-standing connection with us as well. So yeah.
Yeah, I could talk a little bit about partnerships. I think the way that, at least for me, partnerships have developed is, I don't know if this is working, but anyway, if everyone can hear me. It's a very organic process, and it comes out, for me anyway, and it comes out mostly from collaborations. Because in what we do, for example, when you're looking at invertebrates, you're looking at many different groups. And lots of times when there's an effort to look at the fauna of a particular area, tackle a particular problem, the specimens that are requests for identification and so on often go to people who work in different groups, so specialists in different groups, and then the way we come out of it usually is that then we probably, a lot of us, at least me and Gavin, for example, probably have different partnerships in the same institutions, with the same institutions, but coming out in a different way. Moss Landing, for example, a lot of the tsunami work that I do is in collaboration with them as well. So that's how these come about. And then from that on, in terms of funding, in terms of who does what, where, and then those usually come, again, in a very organic fashion, rather than, approaching someone and say, hey, how would you like to partner with us? It doesn't really work like that for me. I mean, it's not to say that we don't talk to people about the possibility of collaborations, but it's a very organic process.
I was going to say that I think that is loud. Also, for those of us who did our training here, undergraduate and graduate work locally, we just naturally, through the process of understanding the agencies that are responsible for especially tracking rare species and keeping lists of and managing all the flora and the fauna in the province, we just naturally start to understand who's taking care of what, and those relationships develop. Yeah.
Yeah, it's a big networking exercise, and we all know hundreds and hundreds of people everywhere, so it all develops, as Henry said, sort of naturally.
Sure. Yeah, so I mentioned 27 people that collected tissue for us. So those are not like a really formal partnership, but every spring there's this circulation of emails that goes around the Northern Hemisphere saying, where are you going in the field and can you collect tissue of X species for our research? So I've been collecting samples for a guy in Japan, a guy in Bellingham, and a group in Oslo. And that kind of, you raise your profile by publishing and attending meetings, so that's how people begin to know what you're doing.
And the reputations are sort of built on that. And a lot of these organic partnerships and invites to BioBlitzes are because people know, oh wait, there's good spider people at the museum. Make sure we get in contact with them because we want to have them at this event or to speak at this conference. There's who's the fossil person on the coast? Okay, that's Marji. Contact her. And you kind of get labels put on you, but in the best possible way. That labels of, wait, RBCM has a good collection of this, I think. And the more we get things available online and aware of our collections that are there, then you will get those contacts. People can quickly look and say, wait, they have how many dragonflies? I should probably contact them to find out about their data and talk to the people that are there. So reputations do come in even before the partnerships.
Yeah, this might be a good moment for me just to emphasize that you see a number of dynamic and productive curators and collections managers here among us, but we also have a very strong network of research associates who add to the power of our research immensely. You heard Claudia talking about Rob Bennett. We have Richard Hebda here. We rely on them to expand the amount of plant and animal groups that we can look at, that we can study, and they also bring connections from all sorts of places, all sorts of people around the world to the museum's research group. So we couldn't do what we do as well without them.
I'd like just to add a comment about paleo, and because we're covering all of life, we have to go to experts around the world, and that's probably why you saw so many contacts there. So we find, when we make a new discovery, we find the expert globally, and it also, paleo and earth science knows no boundaries for that we see traditionally in our political world. And so we have to cross those boundaries and understand that, especially because of the plate tectonic processes of the past. And we're talking over 540 million years that we have of complex life that we're studying here in British Columbia. So we have quite a diverse of experts that we go to. And I just want to add, we also make use of industry because they're interested in understanding the geological evolution and the resources of the province.
I should also say, in terms of research associates, it works the other way around. So many of us are research associates in other institutions as well. For myself, for example, I'm a research associate at the Royal Ontario Museum in invertebrate zoology and invertebrate paleontology. So it goes both ways, this exchange of knowledge and expertise.
Hello, I have a question probably for Claudia and Joel. So imagine down the road your documentation work has identified some insects or spiders at risk, and that has led to some maybe some actions to help protect those, which probably has to relate with habitat loss. So maybe it's policies trying to encourage people to reduce urban development or to manage livestock in agricultural areas better to protect those. What are some key messages that our team of people in learning or our team of people in marketing could use to help convince farmers and people developing properties that this spider matters?
Well, I'll tell you about a couple of the rare spiders. And one of them you'll see we may not be able to do very much for. It's at the northern extent of its range, and it just lives at Stag Leap Provincial Park. And we really did survey quite thoroughly around it and did not find it anywhere else. So, and it's a little tarantuloid, so it's relative of tarantulas, and it's 3 millimeters long. So no one's going to find it. It was hard for us to find it, right, Rob? It's a very difficult spider to find. And it lives in higher elevation area that isn't, unless the only thing we can talk about is climate change in regards to that species. Another one is the bog spider, so the Georgia Basin bog spider, and that species does occur in some protected areas, including Burns Bog. So there are habitats that are already set aside that it happens to still live in. So that's a good news story in some ways, and then in other places where it's been extensively surveyed for, have already been changed enough that there isn't really much, it might not recolonize. So it lives on Tumbo Island, which is a protected area, part of the Gulf Islands National Park. Also at Island View Beach, which does need a bit of help with sea level rise. So.
It is a challenge that we have, and I think this is the best, means we have, is people that are great at speaking, that have great stories to take them to the public. I mean, we have a built-in bias. I mean, even the Luvar is more charismatic than most of the things we try to present to the public. I mean, we have an initial bias against what we do. I think the Luvar is not that attractive compared to the spiders and the flies, but I know I'm not the norm in the public. So
I like the spiders.
It isn't. So, I mean, we have that, and I think the best thing you can do is to sell the real life history, or sell the evolutionary history, or sell what are these things doing. It helps when you get good photographs. I mean, I went to Shane for our pocket gallery and said, find a way to take a great picture of this, and we'll have some nice landscape. And then you can sell it to the public. I mean, it's really hard to sell it to Kosiwig to get them protected in the first place. But like you said, then the public has to believe you. Why should I really care about protecting this bog for some stupid little fly? And that's true, and we have to do a good job of selling it. It is possible. I think the encouraging thing is you can talk to the public and get them over the fact of, you know, their own internal hierarchy of what's a valuable animal and what isn't. It is possible, but stories help.
Maybe Richard has the answer.
There are many answers, but first you need to know what you have. You know, you can't talk to somebody about something that you don't know what it is. Second, you need to give it character, give it, whether it's charismatic or not. Is it unique? Do they have something that other people don't have? Give it special attributes, which many of these things have globally. The work that you're doing, the work that we have done. And then you kind of gather the information. Facts actually matter. People pay attention to facts unless they're, you know, the spawn of Satan evil developers or something. They don't care. But give them information and say, you know, and we did this, and we mentioned Burns Mob. When we did the work to try to preserve it, we had people come from around the world, and if people come from around the world, we had a man from Germany who said, see that big bridge in there? He's talking about the Alex Fraser Bridge. In Germany, they would rip it out. It wouldn't be there. So we need, and we have those connections to provide perspective. You know, I look back at the years, Rob and I've been here working a long time, that only now do we recognize and try to make the point of how special British Columbia is compared to the rest of the world. And your presentations demonstrated that. Holy smokes, you know, let's stop twiddling our fingers about some little spot in the Alps, and let's really talk about where the hot action is about global northern biodiversity, which is British Columbia, for God's sakes, and we barely even know that.
So good to have you back, Richard.
I'm letting him go.
The other thing is you can take that information, if you have that knowledge, and make sure it's shared within the government and otherwise, and they'll come to you, as they did to me in the case of the Maccabee fossil beds, and say, you know, is this important? I mean, you have status. Yes, it is. And here's the factual information. I've got it lined up here, presented. Even government guys figure this out eventually. or you can act as the conduit for others, because you have a status, to places. And then what they do, and this is what happened to me, and you know, it's a bit, I should keep it a secret, but basically they said, look, you made the point, this is really important, you've talked to the people, here's the pencil, draw the area you want protected. And then you look at them and you go, serious? Yeah. And I go, like the fossils? No, all of the values. Like the cliffs and the rare species, yeah, all of it. You got a pencil? I got the pencil. Drew it. Protected.
That was the whole province you drew the line around?
Facts, knowledge, make sure people know about it, and you spread that information. Show them how special it is from your perspective with your enthusiasm. Bring in people from other places who back you up and say, you know, this guy's not blowing smoke, he's not some kind of loony bin environmentalist, which is what people often think. No, reliable, reputable, passionate, it'll work, but we have to get out there. So we have to know and be certain what we know, and then we have to get out there and make sure the public knows about this, both at the collective level and at the individual level. Talk to the farmer. You'll find how much farmers actually really care about their land, or most developers really care. They're not, as I, you know, I was a little bit extreme there, but most people will if you come and talk to them on a one-to-one basis. I gotta go on.
Thank you, Richard.
Thank you for your rant. Love the rants.
Yes, I did originally want to ask about how you name these things, especially in the light of spawn of Satan and so on. That sounds very interesting, Gavin, to explore. But at the moment, I would like to ask, about the question of the creatures on the debris, the species on the debris, and how you deal with the possibility of their affecting the native species here. I mean, is it a long-term job? I mean, you must have to be very careful, because it must have been happening naturally up to a point already, but now that you've got this excessive amount coming through, how do you deal with stopping any problems that are going to occur because of them?
Okay. Okay, I think let me just repeat the question. I think the question is how are we dealing with all these organisms that are coming in on debris in terms of their potential risks of invasion, right, versus what's already here? Am I all right? And you're right. It is a long-term process. Now with bioinvasions, often there is what we call an invasion lag, in which you can have non-native species. Now, not all non-native species are invasive species. No, but what you have are, you can have species that are potentially invasive in an area for a long time before, they could be here for a long time before they are detected, before we actually see them, know what they are. Or B, they could be here for a long time before conditions are right for them to then proceed in terms of an increase in numbers and having an effect on the environment. One example I can think of is mitten crabs, right? The mitten crabs, they live in estuaries and brackish water. They have to survive in normal marine. Now, in the UK, for example, going back probably 60 years, the occurrences of these mitten crabs in the normal shoreline areas, they were there for 60 years before they had a series of droughts, which then allowed the mitten crabs to then move up inland into different habitats where they then flourished. So that's one way in which these things could happen. Now, it translated in terms of the tsunami debris. As I've shown you there, now there's some of these organisms are more easily recognizable. And when I say more easily recognizable, I don't mean that they're necessarily, well, what I mean by that is that, for example, with fish and sea stars, now there were invasive sea stars on the debris, and those were removed pretty quickly. And to some extent, some of those groups are already established here. Now, but there are other organisms on there, and most of the organisms on there are very, very small. Now, because of the nature of this transport, and these are coastal organisms, and these are coastal fouling organisms. So they're organisms that are living, normally they're living on other surfaces, and then living on the debris, and then they have other organisms that are living on them, and in turn other organisms living on them and in them. So they're very, very small and hard to detect, right? So that's one issue here. Now, and that boils down to back to why we need taxonomists, because you have specialists in different groups, and a lot of these groups are not well known. So not only can you normally not see them, they're easy to miss, but when you do see them, you don't know what they are. Okay, so in terms of their life histories, in terms of their identifications, in terms of what they're going to do, so all those questions need to be answered. And then, so we have 289 to date Japanese species on the debris. Now, how many more have we not detected? We don't know. So, when I say yes, there is a risk of invasions. Has it occurred? Well, we haven't detected any at this time. That doesn't mean there is no risk for that, and that doesn't mean it hasn't happened or won't happen. We just haven't found that. So that's a long-term issue. Now, right. So in terms of EDRR, so rapid response, early detection, rapid response, right? What does this mean for EDRR? So the more you know about what the organism is, and its identity, what it does, then the faster it is that you're able to respond to it. right? Because you have to be able to spot it, you have to be able to respond to it. So what does that mean in terms of EDR is that the bigger species are usually, in terms of the kinds of lists that are published and the kinds of efforts that are going on for this rapid response early detection. And a lot of these are larger species, species that you can actually see and pick up, and not so much for the smaller organisms. So now that's why it's important for this work to be going on, because not only do we need to know what they are, but in order to know what they are, we need to know what we already have and what we're looking at in terms of distribution of these organisms up and down our coast. And we don't know that. Or even global distributions, we're beginning to sort those out as we're going through this process. So this project, it's not only, when we're working on the tsunami material in terms of the systematics and taxonomy, we're not only talking about what is there, but we're also looking at a bigger picture of how organisms are distributed and what does that mean. And in some cases, it leads us to reshuffle what we think we already know about global distributions of these species.
Thanks, Henry. Question at the back, ma'am?
I was just wondering about, for Marji, I was wondering about, for dating the fossils, why did you use strontium isotopes? Don't you usually use carbon-14 because it's like more abundant? What are the advantages to using strontium isotopes?
That's an excellent question. The question's about using strontium isotopes and not other isotopes. This is the match for the shell material of the microscopic fossils that we were studying, and the strontium is in that shell material. And it's also the age range that we're talking about. And the curve, which isn't much of a curve, it's more of a straight line for strontium isotopes, gives us more precision. And there's only a few little areas on that line, actually, that deviate from that. So it gives us very precise dating, and it worked excellently. The disadvantage of it is that if the rocks are altered in any significant way by thermal conditions, heating, or that sort of thing, then it doesn't work, it gets overprinted. But that was our example that we found in the Comana Group strata from basically Barkley Sound down to Souk, that the alteration is very low, so that we can actually succeed in getting the appropriate strontium isotope dates. And we can also use that in comparison to the actual taxa that are recovered too, and we know that they come from that approximate age range. So we don't have to do as many dates, because now we know what these taxa are, and they're in this certain age range, and we can use them without doing the dating by looking at the stratigraphy in the area. So excellent question. Thank you for asking that.
The other part of that is that because of the half-life of carbon-14, you can't date objects older than about 50,000 years. They just decay because of the half-life. It's gone.
So 55 million year old fossils are no good for carbon-14. Ma'am, at the back, you've got a microphone now.
Thank you. It's to do with, and you may have partly just answered my question. You're mentioning indigenous versus invasive species. Is there a benchmark timeline to designate things as indigenous, because maybe they were invasive at some point? And also the second part of that is, do you ever concern yourself about the fact that you're finding very rare species and you're identifying them possibly as invasive when they may actually have been here and just never seen before?
It's not
Go for it.
It's like the definition of a weed is something you don't want there. The definition of invasive or introduced is you just saw it there. So it's really a subjective term, and it's something that you really, reading literature, you interpret it how you want to. Now, some things that are a problem, some big new forest pest that's wiping our trees that was in Europe and was never seen in North America before, yes, everyone's going to call that invasive and a problem and try to control it. But one fish that shows up and we've never seen it, you could call it invasive, you could call it new introduction, you could call it never previously detected. Those terms are meaningful depending on the questions that you're asking. So we ask them, but sometimes we don't spend a lot of time thinking about it if it's not the question we're asking. So, for example, the Japanese mosquitoes, Aedes togoi, I reared them out of tide pools, and they're here. And I looked, and Richard Ring, who is a research associate of ours at University of Victoria, found them in the 1980s, was rearing them out of ponds around Vancouver and here, and said, no, these are likely new. And it's a concern because they could possibly be vectors for Japanese encephalitis, and these are probably newly introduced. And then other people said, well, no, we found them in other places on the coast. They may have been introduced thousands of years ago. They may have always been here. So sometimes it's a difficult question to answer when it showed up, and it does matter, but sometimes we don't care.
Ken?
This highlights the value of collections. So we're very fortunate, at least in botany, that in the late 1800s, there were some very good botanists that documented a lot of plants that are already here. So if there's no record from that time, and there's a record from the 1940s and 1950s, then conclusion would be that it's probably not a native species, it's an introduced species. It gets tricky. Henry and Joel and Gavin and I were just in Richmond at an early detection rapid response boot camp, and I talked about a particular grass that has two genetic types. There's a North American type and a European type, and they cannot be distinguished very easily based on appearance. You need DNA markers to distinguish them, in which case they're readily distinguished. So land managers have this dilemma. They have this grass growing in their wetland. Is it the native one that they should leave alone, or is it the invasive one, which has been tremendously invasive in eastern North America, and do they need to control it? So it gets complicated.
Yeah, so I can add to that, actually. I think, that's a great question. Now, in terms of, you notice that I often refer to non-indigenous species as opposed to invasive. And I always say potentially invasive. Now, in terms of what their effects are on the existing ecosystem and what they're doing. So, specifically in terms of the Japanese tsunami debris material, in terms of non-indigenous species, we know in some cases we were able to, we were not always able to get usable DNA, but in some cases, so we're two main lines of evidence in terms of the species identification, and one is morphological, and the other one is molecular. And in many cases, we were able to put both of them together, for example, and identify the organism back to the source population where the tsunami occurred. So that's one way something is really it's not from here in terms of a specific population. Now, the other question, the other part of that question is, well, how do you know how long something has been here, and what is the cutoff point? There really is no cutoff point. As Ken was mentioning, these records of historical distributions are very important, and we're constantly refining our knowledge of distributions and what we consider to be endemic versus non-endemic, for example. Now, we know that in the area that I work in, I work on hydroid taxonomy, hydroid zones, and we know that in my area, for example, that from working with West Coast hydroid zones, I know that the level of endemicity is higher than we normally think, because in those cases, there are lots of groups that are commonly thought of as being cosmopolitan, when they're actually species complexes, and they don't represent any kind of real individual groups. So those kinds of definitions are constantly being refined. So we're constantly learning, getting a better picture of what we think is here. What is that picture? How long has that been here? And in terms of introductions, and things get discovered and rediscovered. Now, we've described species, for example, that have not been seen, for example, since the 1930s, and then we see them again. And without the records, then you have no way of knowing when the last occurrence was. So those things those kinds of records are very important, and that comes back again to everything that we do in the museums and on a daily basis is identification of organisms. Is an exercise not for its own sake, but because it enables us to better answer questions like that. So yes, so when I talk about indigenous versus non-indigenous, the non-indigenous should not be taken as a synonym for invasive.
Axel?
Thank you for these wonderful presentations. In your presentations, you focused on organisms that are relatively large. I think the smallest ones were a few millimeters. But biodiversity extends to a much smaller size. Could you comment on what they're doing with respect to organisms at the microbial level, for example? Because they are part of that continuum, and based on what I know, they're a really important part of the continuum.
I can comment on that from the perspective of having recently done an environmental scan of natural history museums, 10 of them around the world and locally, to find out what they're doing that we're not. They're doing microbes. So big museums in Europe and in Australia and even some in Canada are now taking water samples and soil samples and keeping them in cryopreservation for the study of microbes over time. I think that is a frontier that is rolling through some of the bigger museums in the world for sure. We're not there at this point. That being said, a lot of our specimens in the botany collection, for example, and I'm sure it's true in other collections, have samples of soil on them for botany. So just by accident, we sometimes end up having soil samples in our specimens. It's an interesting question, and I think we're going to have to look at whether we want to be doing that.
Just one other comment about that in terms of, and you're absolutely right. I mean, the organisms below ground probably have far more impact than we realize, or we probably do know. But just the identification of these organisms has become much more maybe easy, in the sense that in the past, to even detect their presence, you had to culture them. Now you can just take a teaspoon of soil and sequence the DNA of that teaspoon of soil, and you will get DNA sequences of organisms that you don't even know what they look like, but you can identify them based on that.
One of the other things that we, with the vertebrates, we don't pay enough attention to is their parasites. So when you get a luvar from Baja, what came with it? Like, we're all colonies. We're not an individual. We have bacteria. We have parasites. The luvar actually had a copepod attached to its eye. And this copepod was about the length of my hand, hanging off the eye of this fish. So that specimen actually went into the collection. But the actual, like, blood-borne parasites, flatworms or nematodes or anything small like that, we really don't adequately sample that. And that also is part of the diversity that's here. Yeah.
Well, I'd certainly like to comment about this because I started off as a micropaleontologist. So yes, and in terms of the paleontological record, micropaleontology, like the study of single-celled organisms like the foraminifera I showed you on the slide there, ostracods, which are multicellular but very tiny, these are all less than a millimeter in size. The pollen that Richard was studying, dinoflagellates, these are all sort of the foundation of the food chain, and they come in huge numbers, and so they provide a wonderful statistical basis from which to study life. And here at the museum, aside from Richard's pollen samples and a few small collections of microfossils, we're really deficient, and this could be a huge area of growth if we choose to do this. In terms of the Geological Survey of Canada, who has studied a lot of the microfossils, they are the foundation of the knowledge of our stratigraphic record out there. And because this was also driven by industry, because in those days oil and gas was a big component, and they were small and you could study them within the drilled material that they went down and studied. So they have formed the foundation of a lot of paleontology, and then we use the macrofossils to tie in with that. So it forms a multi-system biological understanding of the stratigraphic record, the history of life on this planet.
Yeah, in terms of the tsunami material the study, we are. You're absolutely right. And again, as I've mentioned, that it's on the smaller end of the spectrum that the sampling. There's challenges with sampling and challenges with identification. So, but we actually, a lot of what I do in terms of the hydrozones also grade to the smaller end of the spectrum, we, in terms of things like parasitic and intramuscular parasites. So some of these hydrozoans are parasitic, and so we're looking in. We were able to detect, for example, in muscle tissue, some of these parasites, and that's where the molecular work came in very useful. So we do look at groups like that, but you're absolutely right. So a large part of when we're going back and estimating in terms of our sampling, what is it that we probably have missed the most, and that end of the spectrum is where we're lacking in terms of being able to detect and identify these organisms. And part of that also is because we don't have anyone working on specific groups. It comes back to that a lot, in that sometimes you see this bias because there are people working on specific groups, and sometimes because they're so hard to detect. And when you're looking at a mass of biological organisms and a collection of material, I mean, it's extremely difficult to try to tease everything out, and to try to tease everything out in time where you're getting usable samples.
Could I make a point?
Rob, you have the last question of the
just
day.
with, it would be nice to do full-scale biodiversity research. It's practically impossible. It can't be done. Certainly not with the funding that the Royal BC Museum gets. The Royal BC Museum does a pretty good job with the groups that we do work with, but most of us are familiar with the biodiversity triangle. 5% of at the top of the triangle is where 95% of the funding and research interest goes. The remaining 95% is pretty much unfunded. 75% of all the things on the face of the earth are arthropods. We have 2 people here, 3 people working on arthropods. It would be beautiful to be able to do microbes, but that's another part of the foundation. How are you going to fund a program like that at a regional institution like the Royal BC Museum that does a truly fantastic job with the groups that they do have curators working with? Thank you.
I think it's lunchtime.
It is lunchtime.
Thank you for your participation.
We'll see you back at 1 o'clock.